Hello students, once again welcome back to our biology class. Myself Dr. Ravindra here. So in the earlier class we discussed about number of important things concerned with the biomolecules. Yes, we discussed what do you mean by biomolecules and what are the microbiomolecules as well as the macrobiomolecules. We have discussed it. Fine. Yes, so looking into the types of biomolecules, yes, basically they can be proteins, carbohydrates, lipids as well as nucleic acids. Fine. So we find these biomolecules which are dissolved in the cytoplasm and they are easily available in the acid soluble fraction. There are number of biomolecules, yes, they can be found in the acid insoluble, we can say that fraction, and they are macromolecules. Their molecular weight will be definitely higher. Fine. So we studied about basically, yes, the what you can say, mainly the structural units of biomolecules as such. We are discussing the structural units of biomolecules as such. Proteins are basically made up of what? Proteins are basically made up of amino acids. Yes, we have studied that proteins, yes, they are also called the polymers of amino acids. So mainly what we can say, they are the polymers of amino acids. So amino acids are attached to one another mainly by the polypeptide bond. So yes, we have studied that, and we have also studied essential as well as non-essential amino acids. Essential amino acids are those which are not synthesized inside the body. We need to obtain it through the food or through the diet. So non-essential are those which are synthesized inside the body only, and we need not to consume it too much from the outside. So they are called non-essential. Yes, we have studied. And we also discussed basically the carbohydrates are made up of what? They are made up of the simple sugars. Yes. So in detail also we will discuss. And then lipids. So lipids have lots of significance in our body metabolism. Yes, the fat is essential, that can be acting as a reserve energy. The fat may be acting as an insulator, yes, around the nerve fibers. And fats are essential for mainly number of metabolic activities as such. So the fats are basically made up of what? They are made up of glycerol and fatty acids. So today we will discuss about the structural units of the fats, structural units of the carbohydrates as well as structural units of mainly the nucleic acids, because already we have studied mainly the as such the proteins, and then the protein structural units are called as amino acids. Amino acids can be acidic, basic as well as neutral. We have also studied in the earlier class zwitter ionic form of the amino acids. So that also, yes, how actually they balance the positive and negativity in the amino acids. We have studied all that. So today we will be focusing more on mainly as such the lipids as well as carbohydrates, and then we will be studying about the nucleic acids. So looking into the basic type of lipids, as in the earlier class we discussed, the lipids are made up of either 16 carbon also, or they may be 20 carbon. 20 carbon example is arachidonic acid, and 16 carbon compound is mainly palmitic acid. So let us study the structural details of these, mainly what we can say. Yes, we will study about as such the structural details. So first let me just give you the structure of as such the, yes, units of lipids mainly. Yes, there may be few examples. So lipids, so made up of fatty acids. We have discussed the fatty acids are unsaturated also, saturated also. Unsaturated are good for health, saturated are not at all good for health, because unsaturated they have the very low melting point, easily digestible. Saturated, yes, they have the high melting point such as animal fat. So they are made up of fatty acids and glycerol. Well, so they can be, lipids can be 16 carbon compounds. 16 carbon compound as such. The example for this, palmitic acid. Palmitic acid. So, considering a palmitic acid structure, it is basically the CH2, COOH. So, the CH2 by again multiply by 2, so we can find the 2 carbon, and here we can say that it is in fact 14. So, 14 carbon, so 14, 15 and 16 carbon compound, palmitic acid. So let us understand simple, yes, what we can say, the lipid molecules such as the glycerol. So the first one what we discussed, 16 carbon compound palmitic acid. Second compound we will discuss as such, mainly it is the glycerol. So let us understand. So this glycerol, well, I hope you are able to see this. Yes, the second one, yes, we are going to discuss glycerol. It has CH2, CH and CH2, all of them with the OH group. So this is what we called as a, yes, simple structure as such, glycerol. This is the glycerol. Well, so I hope the screen is very clear now. Yes, what you are observing, glycerol. So this glycerol can be, yes, converted into triglyceride also. How? So glycerol, yes, when attached with, we can say that addition of COOH, side chain R1, COOH R2, then COOH, the R3. So here what you can say, the OH group, these, and the one H over here. What happens further? Yes, this leads to the formation of triglycerides. So the third compound, yes, let me tell you, it is CH2, CH2. So it gives us what we can say that COO, yes, and then it is the R1. So it can be written either in this form also. Instead of like this, we can write, let me mention here, it can be oxygen, carbon, double bond O, R1. So oxygen, carbon, double bond O, R2. So oxygen, carbon, double bond O, and then R3. So now it is what we can say, a triglyceride compound. So triglyceride plus what will happen, 3H2O. So release of the 3 water molecules, means these H and these OH, 3H2O is released, and ultimately it is going to form what this compound we call it as a triglyceride. Triglyceride. So this is what we can say, the third important compound. Yes. So a glycerol molecule can be getting converted into triglyceride. Yes, when there is the addition of carboxyl group COOH and the variable group R1, R2, R3. So this is a glycerol, but addition of COOH and R1, R2, R3, that leads to the removal of OH and one H. So ultimately release of 3 water molecules and then formation of yes, triglycerides. So this is what we say, the triglyceride. Now we need to understand further one more important example, number 4. It is the lipids can be complex. Lipids can be much more complex. They can be what we can say, the combination of various groups, we can say. Best example, it is mainly phospholipid. Phospholipid, yes, as such. It is mainly also called as a lecithin. Lecithin is abundantly available in the egg yolk, and that is very essential for a number of metabolic activities, and also yes, for the nervous system, lipids are important because the lipids forms the outer coat, or even they are forming the myelin sheath around the nerve fibers. Well, so the phospholipid as such, why? Because it has a definitely the phosphate group. Now here as such, definitely it is having the glycerol group CH2, CH, again CH2. So this has mainly what you can say the oxygen, and then the carbon double bond O, and then the R1 group here, and then here what we can say mainly the CH. So it has the oxygen, carbon, then the R2 double bond O, and then here what we can find the oxygen, and then we find yes mainly the group as such, it is the mainly yes we find one more oxygen as such. Yes, we find the oxygen, fine. So here we find mainly the OH group, and then we find mainly the phosphate group double bond O. Sorry, this is also an oxygen what we can find here, CH2. Yes, it is the OH. Here it is the oxygen we find CH2 here, one more CH2, and that has again the nitrogen, and again what we find CH3, and CH3. So this group indicates mainly, this group indicates ethanol amine. Ethanol amine, there are 3 CH3 group, so trimethyl group. Okay, so as such, here we find this as orthophosphate group, and yes what we find here COO on the either side. These are the ester groups. Okay, yes, so this CH2, CH, and the O, yes what we find it is mainly glycerol. So with this kind of yes complexity, it is called as a phospholipid having a orthophosphate in it. So this is very essential that you need to know how actually the compounds can be much more, it keep on becoming complex, what we call it as mainly the phospholipid. So this is very essential as such, you need to know. So well, that is about mainly the what we can say, it is the orthophosphate. In fact it should be, let me just clarify this, it should be the O is here, yes, it should be the O directly, and OH should be here, yes. So this is what we call as mainly the compound as such phospholipid, which is very important, and it is a complex lipid also. Fine. So that is what we say mainly the ester groups, glycerol, orthophosphate, ethanol amine, trimethyl. So these are what we say mainly the phospholipid or the lecithin. Other than this, one more important lipid is there. It is called as mainly the cholesterol. Cholesterol, yes. So it has a particular kind of the multiple ring structure, what we can say. It is having, yes, the ring structure such as A, then it has mainly the B, yes, the ring structure as such, mainly it is C. Imidazole ring, what we say, this one. Okay, D. And then it has one more important structure as such. So here what we call it as mainly, yes, it has the ring structure as such. Yes. So this is mainly the carbon as such. So somewhere around more than 20 carbon compound cholesterol. It has more than 20 carbons. 20 carbons. And so here what we can say, this ring, peculiar ring structure is called as cyclopentano. Cyclopentano hydroxy phenanthrene ring. Phenanthrene ring structure. This is what we talk about the cholesterol. Cholesterol as such, it is the most important precursor. Cholesterol is a precursor for various kind of what we can say mainly steroid hormones. What are those various kind of steroid hormones? Such as cholesterol is essential to make up the androgens or testosterone. Cholesterol is essential to make up the what we can say mainly the estrogens and progesterone also. Well, so cholesterol, let me mention here, cholesterol is the precursor for testosterone, then even for estrogens and progesterone. So this is all about what we have discussed in the detail. Yes, mainly the structural units of the lipids, what they are basically made up of. Hydrocarbon chains only, yes, they are made up of. But we can say that a carbon and hydrogen can be more a long chain as such. 16 carbon, yes, CH2, 14 COH, yes, it is a 16 carbon compound palmitic acid. Glycerol, CH2, OH groups for each carbon. This is a glycerol, but when it is mainly attached with the three side chains, the carboxyl groups and R1, R2, R3, then yes, it forms mainly the OH group and one H from the carboxyls removed. Ultimately, it is a reaction of dehydration. So yes, dehydration happens, removal of water, dehydration or removal of water molecule, ultimately going to form CH2O and COH group. So there are three ester groups we can find here as well as the R1, R2, R3. So this is what we call as a triglyceride molecule. And then, so it can be more complex lipid as such. Phospholipid, best example is lecithin. So what you are finding here? So two ester groups you are finding, and then one glycerol group you are finding, one orthophosphate you are finding, one ethylene amine trimethyl group you are finding. This is what we talk about the lecithin, which is abundantly available in the egg yolk. Cholesterol having a peculiar structure, 20 plus or more than 20 carbon we can find in this peculiar ring structure, what we call a cyclopentano hydroxy phenanthrene ring, and cholesterol forms the precursor for testosterone, estrogen as well as progesterone. This is all what we have discussed about the mainly regarding the lipids as such. So let us go ahead mainly. What are the important structural units in case of the what we can say mainly the sugars? So sugars are the mainly carbohydrates. So structural units in the carbohydrates, what they are basically made up of. Structural units in the carbohydrates are mainly the sugars, what we can say. These sugars can be as such so simple. They can be the 6 carbon compounds, so we call them as a yes hexoses. They can be 3 carbon trioses, 4 carbon they can be tetroses, and then 5 carbon they can be pentose sugars, we can say. So yes, so sugar molecules are not only restricted in the what we can say the carbohydrates. Yes, we can say that sugars also present in the DNA as well as RNA. In the DNA, deoxyribose sugar. In the RNA, it is ribose sugar. Let us understand now. So, carbohydrates. Structural units are sugars. Sugars are glucose, we can say. Fine. So the carbohydrates, we can say that mainly with the 3 carbon they are trioses, 4 carbon tetroses, 5 carbon pentoses, and then 6 carbon hexoses. Hexoses we say. So to give an example for an hexose, yes, we can say that for pentose, yes, it is what you can say ribulose sugar, and then for hexose it can be the glucose. Okay, so ribulose sugar can be, yes, its molecular formula C5H10O5. Glucose formula is C6H12O6. So let us understand basically the hexose or the glucose structure. Yes, the glucose basically having the terminal oxygen and then alpha carbon. Yes, here the OH group is here, OH group is here, and then we can say that CH2 will be there. So the first carbon, second, third, fourth, fifth, and the sixth carbon here. This is what we can say mainly the glucose or the hexose. So comparatively, when we talk about mainly the pentose, so this is what we say that mainly the hexose or the glucose, the structure what we are observing. So glucose, okay, plus what we can say fructose. When they combine, it forms what we call as a sucrose. So sucrose is a disaccharide. Disaccharide means, yes, the glucose plus fructose can become a sucrose, and then glucose plus galactose, that can form mainly lactose. So it is also what we can say the disaccharide. So two different kind of glucose, yes, glucose and fructose, glucose, galactose, all are having C6H12O6 only, and these are also what we can say mainly the sucrose can be mainly in the sugar cane, in sugar cane juice we can find it. Lactose, yes, in the milk we can find it. So thus whether the carbohydrates seen in mainly what we can say either plant material or sugar cane juice, yes, they are made up of the glucose only, or if they are there in the animal material or in the milk such as lactose, yes, they do have the glucose in them. Thus variety of sugar molecules basically they are constructed or formed by glucose only. So the monopolymer as well as we can say that homopolymer and heteropolymer means same kind of sugars are forming the long chain of sugars. We can say that it is a homopolymer or homopolysaccharide we can say. So thus we can say that sugars can form homopolysaccharide as well as heteropolysaccharides. So yes, what you are observing, the monosaccharides means mainly the glucose or the sucrose or the fructose. They are all monosaccharides, but when they combine, so different type of sugars together, yes, it is called as a heteropolysaccharide. Now it is a 2 units of sugars disaccharide what we are observing here. So sugar molecules as such, we can say that mainly the sugars or the glucose can form long chain, and that becomes what we can say that the heteropolysaccharide, and it can become homopolysaccharide. So here heteropolysaccharide means a different kind of sugars. Homopolysaccharide means a similar type of sugars. Well, so this is all about what we have seen regarding the glucose, and it's going to be found in what kind of different sugars, yes, we have seen. Let us understand now the pentose, okay, or ribulose sugar. So yes, the ribose sugar is an example of pentose only, and that can be, yes, ribose sugar in RNA, yes, it is found. It has the structure, there is a terminal oxygen, yes, the carbon, alpha carbon we say here. So it is the OH group here, OH here, OH here, and OH here also. And then what we can say that, so the first, second, third, fourth, and CH2, yes, the fifth carbon is here, okay. So thus they are having mainly 5 carbons. Well, so this is mainly seen in the RNA, but so we can say that 2-deoxyribose sugar in the DNA, okay. What is the structure? So terminal oxygen, and then the first carbon, yes, second, third, fourth, and fifth, like this. So here it is OH, here it is only H. Oxygen is removed, okay. That's why it is called the 2-deoxy, means at second position oxygen is removed, and then here it is again OH, here it is OH, here CH2. So this is what we talk about the deoxyribose sugar that can be seen in the DNA only. Well, so this is all what we talk about regarding the sugars, which forms the mainly structural units of what we can say the carbohydrates. So we need to understand one more important concept as such. Structural units found or seen in the mainly DNA also we will discuss. Then we will go ahead with the what you can say physiological role of proteins and their structural details. Then we will discuss about the polysaccharides and some important what we can say homopolysaccharides, heteropolysaccharides, and their physiological importance. Then mainly these what we can say long chain of sugars, how they are connected. They are mainly linked by the glycosidic bond, that also we will discuss, okay. So we are going to study even about how the DNA double helix and what are the structural details of the DNA. Yes, all those are the very essential and important concepts. Well, I hope you got a clarity now about this here. Carbohydrates are mainly having the structural units. They are called as the glucose or the sugar. So these carbohydrate units can be 3 carbon compounds, trioses, 4 carbon, tetrose, 5 carbon, pentose, 6 carbon, hexose we say. For hexose, best example C6H12O6, that is the glucose molecule. So this is the structural formula of the glucose. So glucose plus fructose both are having C6H12O6 only, but different sugars which are seen in the sugarcane juice, and this kind of two kind of sugars, yes, together forming disaccharides. Similarly, in the lactose or milk sugar also glucose plus galactose will be there. So thus, whatever the complex sugars, yes, they are made up of the glucose only. Yes, sugars can form a long chain. We can call them as the heteropolysaccharides as well as the homopolysaccharides. And then sugars can be seen in the DNA as well as RNA. In the RNA, it is a ribose sugar which is having the mainly molecular formula. Yes, it is C5H10O5, whereas one oxygen is less in the deoxyribose sugar, so it will be C5H10O4. So deoxyribose sugar, it has C5H10O4. So here it is a C5H10O5. Okay. So these are the things, yes, you need to remember, what we call them as the structural units of mainly important biomolecules. Well, in the next class, yes, as I told you, we will be discussing some other important biomolecules and their details. Till then, take care, study well. Thank you very much.